Well, dear colleagues and friends, it's high time for us to start this afternoon's seminar. And the speaker is Laurent Guéguen. And I will say a little bit more about him in 1 or 2 minutes, not more. But I should say that Laurent is here within the framework as a scholar, and we are very delighted about that, but he is also, in financial terms, funded by our Natural Sciences program. And I will not go into detail about that. I see my friend Gunnar sitting across here, but it is a history that goes back actually until October, or from October 2010, when I was contacted by a rather prominent Swedish physicist who happened at that time to be the Vice Chancellor of Uppsala and later became the President of the Royal Academy of Sciences. And he wanted basically for us to meet in a group of natural scientists, leading Swedish natural scientists, very much people attached to the Nobel sphere in various ways, and look at what an Institute for Advanced Study could be all about. And I think the plans originally was for the natural scientists who felt some envy towards the social sciences and humanistic scholars to create an institute of their own. They rapidly came to the conclusion, not least after a visit the following year to the Wissenschaftskolleg, that we should rather try to stay together and create an all-encompassing institute. And I've always been a strong adherent, a proponent of that idea. I think it would be absurd if this day we would say that from the point of view of the humanities and social sciences, we don't want to have anything to do with scholars when there are so many fields where we clearly profit immensely from collaboration across those boundaries. And eventually, after many years actually of very hard work, I think I worked more on that than on the proposal when we established Scas once, we proposed an effort along these lines which would at one and the same time respect the nature of an institute for advanced study. That is, every scholar at such an institute should have the same rights, the same privileges, and the same rules as to attendance should apply, but we should also have a component, an element of a more thematic focus in that program. So eventually there were 4 such foci that were proposed. They were very broad, and one of them is the one in which Laurent is working, and that is evolutionary and theoretical biology. Another one was the human brain in its widest connotations, and we had already done a lot of work there since one of our former long-term fellows was Peter Gardenfors, who is a leading scholar in cognitive science. And of course, there are large fields of psychology where you cannot really work unless you're prepared to enter into collaboration with neuroscientists, and the whole field of cognitive science as it is, of course, involves such a transcendence of boundaries. And the third one was measuring, or measurement, the somewhat unfortunate name measurable man. There are other genders who are also measurable, but dealing with the problem of digitization and healthcare, but also informatics more generally. Again, clearly a field that transcends these boundaries. And finally, the theme of exoplanets. And the structure that evolved after long discussions and after rather generous funding from some of the larger Swedish foundations, the Knut Analys Wallenberg Foundation and the Family Erling Persson Foundation, was that we would have one component where scholars would be in residence on absolutely the same terms as everybody else, and one component that would entail that the lead authors of these thematic proposals should have an ability to work on a more long-term basis, primarily in their home departments, and also involving a number of shorter-term visits. And that we made a rough division of the resources along these lines, and we started by having 4 symposia on these major themes, which all went extremely well, I must say. They were exciting events. And we had our first fellows in residence here in the program in the spring of 2018, and they were, as Laurent, involved in questions of modeling, evolution, and theoretical biology. Maria Cervedio and Janet Baumann were here, and then I shouldn't mention everybody else, but you know many of these people, including Troy Day and Suhini Ramachandran, and now you're here. And I think generally, and Gunnar, you will have to correct me if you disagree, but on the whole, this has been an enormously invigorating and stimulating element in the life of the Collegium. And I think the natural scientists, at least with some distance, so, I mean, everybody's positive in the beginning and at the end of a semester, but you should listen to people afterwards. But it's fair to say that all of them have been really enthusiastic about their time in residence, and I think all other fellows have enjoyed deeply this interaction. And the presentations have been generally wonderful, often much better than and more easily accessible than those of people in the social and human sciences. So we are delighted that, Laurent, that you are here, and I think you have already made more friends than I will make in a lifetime. And it's been wonderful. About your background, I should say that Laurent is trained as a mathematician, essentially. He went studied at the École Normale Supérieure de Cachan and in the mathematics section. He got his PhD from Paris, which is Marie-Pierre Curie. He has subsequently worked at the Université Claude in Lyon. And incidentally, we have a sister institute in Lyon, which is called Collegium Lyon, which has actually as one of its foci slightly themes that are not very far from yours, evolution and human brain. Laurent has several major fields of information, so one of that, of research. So one of them is modeling, and more precisely modeling in phylogeny and molecular analysis and evolution. He has also a general interest in algorithmics and path dependency and development, but also then very generally in the whole field of bioinformatics, where he is a noticeable figure internationally. He is a reviewer for most of the leading journals, I think, in this field, and I won't bother you or take up time with enumerating them. He has also made a great contribution by elaborating a database, and I shall not pretend that I know this database. I've read very carefully your application, of course, when you were applying here, and I read some of your background materials, but I'm not, of course, familiar with the details of this modeling effort that you have, the bioinformatics system that you have developed. But you are one of the main authors of this Bio++ libraries, and obviously these are tools for rather highly efficient analysis of evolutionary processes, but also, and these processes include ways of handling linkages in disequilibria and problems of neighborhood dependency. I'm not going to pretend that I've read things I haven't read, but I know that this is extremely interesting, and you're working at, to my mind, as far as I can see, at a field where problems of methodology intersect with absolutely key processes in evolutionary long-term development. So this is a field which, of course, nobody can fail to find a fascination in, and where it's wonderful that this kind of collaboration of informatics, mathematics, and substantial theorizing in a burgeoning field does occur. So you're warmly welcome, and it's wonderful to have you here.
Thanks a lot for a nice introduction. Today I will present to you as a modeling view. I suppose I am a computer scientist, so my point of view is rather about modeling, about phylogeny, the links between phylogeny and population genetics, and where today where the modelings are interfering between both approaches. So at first I will give you an introduction, because as to say that the point is to present the whole field of evolution and different terms I will use afterwards, and then I will come into the more or less slowly into the modeling aspects. So what is genomic evolution? Genomic evolution is the evolution of species as seen from genome. So there are many mechanisms with evolution genomes evolved. And there are many mechanisms that are involved, like I will focus mostly on mutation recombination. We explain later what it means, but there are also many lateral gene transfer duplication when some part of the genomes are duplicated, rearrangements where the chromosomes are mixed and something. And that is the mechanism at the level of genomes, and the main part of my discourses will be that in population genetics, that genomes are individuals, there are 8 populations, and all the dynamics is linked with the dynamics of population, ecological point of view. And so in the population, there are also some population mechanisms, like survival and reproduction of individuals that will carry on the next generation. That is linked with the concept of fitness and the dynamics of genomes that are will be linked with the notions, the concept of drift and selection. So since we talked about genomes, they are made of nucleotides, okay? And we come to the atomistic point of view. We can see first the evolution of sites here. So for example, here we have okay. So for example, here we have a population, many individuals, in which there is one, sorry. Okay. We have many individuals, and each individual may have a mutation, a change in the genome, that will, along with the time, no, that's fine. No. Yes. So here we have many individuals, and around the time, there's a generation by generation, and with the time there is one change in the individual here, and if this change, if the individual doesn't die and can manage to reproduce, is the change in its genome will expand to the population or not. For example, here another individual, and the population here he has he managed to reproduce, but there is no succession in the next generation. Okay, but this is the evolution of at the level of population here, many individuals, and if we consider it at the level of time, we have evolution of population that leads to several species. Okay, that's what we call a species tree, in which there are some different populations, around which we can see there are some variance, some variability in this population. Okay. So the birth of a mutation here, at first we have a mutation. Mutation is a change in the genome, and this mutation brings polymorphism at the level of a population. It means that the population is not homogeneous anymore, containing this feature, what we call an allele, and there is variability that we call polymorphism. And this polymorphism can have several, usually has 2 ways of ending. Either it is deleted, so it disappears. For example, there are many tiny spots here, the white, the yellow one here, and so on. Or I double. I have to. No, not this one. Sorry, this is tricky. Okay. And either this mutation is what we call fixed, so it is expanded by all the population. There is no polymorphism anymore, and thus at that point, this mutation is called a substitution. That will be useful afterwards. So there are 2 features that will drive the evolution of mutation in the population. One first feature is what we call selection. Selection is when
could you either use the microphone or speak up a bit?
So when there is a mutation, the mutation can have an effect on the individual, either good or bad, concerning his environment, the rest of the population, and so the strength, and this is what we call selection. The selection is the feature that will help or prevent the mutation to expand. Okay, so what we call, if there is the concept of fitness, so an individual that is fit is an individual will have a lot of descendants, and so there are the fitness. The mutation can either increase the fitness, in which case there will be positive selection on this mutation. For example, here the blue one, for example, we can say there is positive selection, then good one, good mutation, lot of descendants, and then it expands to all the population. Or it can be neutral, I will come back to this later, or the fitness can decrease, then it is bad mutation, and then usually, I won't come into the details, usually it will disappear because it's not good. And the other tendency that is very, very important is what we call the neutral drift, which is neutral. It is due to the stochasticity of the process. A mutation can a mutation can be fixed or deleted only because by chance. And this, for example, because somebody can be a Superman but can have no descendant. It happens. And the inverse, I won't name anybody. And so in that case, there will be either here a mutation that will be deleted or fixed, but there is nothing, it's not linked with selection. Actually, the two forces are linked. There is drift and selection, and the balance between both is one of the main questions in molecular evolution. And the big feature that is very important in the concerning drift is and what is very interesting for ecologists and people who work on population genetics is that the drift is linked with population size. As you say, this is only stochastic, and when the population size is very, very, the smaller is the population size, the more stochasticity there is, I could say. And so the strength of drift is more important. So for example here, we can say here, for example, there is a neutral, you can imagine here a neutral mutation that goes and then disappears. But here it's the same, but since the population is smaller, by chance, poof, it is expanded all the population. So it's not good, not bad. It happens. Okay? And it can be that even a mutation that is slightly deleterious, not very good, but not so bad, then can be expanded to the population by chance. So there is a strong influence of both selection and population size on the dynamics of the evolution of genomes. So I will touch a little bit of molecular evolution, molecules. So people, when they look at selection, people in biology, in molecular evolution, they are very, very interested into selection, to know what is under selection, positive, negative selection. Specifically, soft genes are under positive, negative selection, and even some sites, people who work on functional molecular evolution, when they want to look at very, very specific genes that they want to see, for example, these genes that is involved in immunitary system, is this amino-acid good or bad, and so on. So they can be very interested in selection in very, very precise spots. And then mostly people are looking into codons, into genes that are the parts of the genome that are expressed to make proteins, and they focus on and as you may say, genes are made of codons, like this one, for example. It's a part of a gene, and in which there can be two kinds of mutations. Either the mutation that will change, so they will change the protein, so the gene will be translated into a protein.
And there are mutations, for example, here, CCA to TCA, we will change the amino acid, that will change the protein, that will may change the function, and then may have an effect, I will say, on the functional and the functional effect. And other mutation here, for example, CCC, CCA to CCG will be synonymous because the amino acid that is encoded would be the same. Okay? And it's very convenient, actually, because on the same part of the genome, more or less long, there are synonymous and non-synonymous mutations. And usually we will consider that synonymous mutations can be neutral mutations. I won't come into the details. Usually we say, okay, it's fine. We say it is neutral. And then on the same spot, we can add both kinds of mutations. And then for this, with the difference, we can measure the strength of selection, because you have neutral and non-neutral. And so, okay, so it is, so this part will be either positive or negative. We have something like a null hypothesis there. So, and the codon, so the many modeling in looking for selection in phylogeny focus on codons, on genes, because of this, and they exploit what we call the genetic code, and all this is possible because of the genetic redundancy. So here is the well-known genetic code, standard genetic code. So as I mentioned before, still a point about the short introduction of evolution. When an individual, when there is a mutation, or anybody can have successors that have successors that have successors and so on, it brings up lineages, genealogies, if you point of humans. And I didn't draw all the lineages, but from here, there you can see there is all the descendants that can go very on and on, okay? And these lineages, if we consider, for example, here, 3 individuals here, for example, B, C, A, it's in 3 species, but I will come back to this later. We can see where they come from, okay? They're ancestral, they're common ancestors, and we can see, for example, here from the lineages that B, C, they may have ancestral here, and the ancestral of the 3 individuals is here, and so on, okay? And what will be important here is that, if we consider the population point of view, is that if we consider lineages, these lineages cannot fit the evolution of the species. Okay, we are used to say, for example, we give more example later, that, for example, human is rather close to chimp, that is, and gorilla are a little further. But actually, we can have some parts of the genomes, or because here that may be closer for the human genome, that can be closer to gorilla than to chimp, okay? Because of the idea of lineages, okay? The fact that some part of the genome can be in this kind of model, and when the ancestor here between A and B is more recent than the ancestor we see. Okay, the thing is that so there is a dynamics of lineages that comes above the main point of view that we can have with only the mutations that go and so on. And after this, I just talked about mutations, but actually, all these mutations are supported by chromosomes. And chromosomes, by themselves, they evolve. Okay? And when there is sexual reproduction or even in bacteria, there will be recombination between inside of chromosome that make, like, for example, here, some parents, if you have some chromosomes here, on the next generation, these two chromosomes will be broken and put together. Okay, for example, here from one gene we have 3 chromosomes, red, yellow, and blue, and they are on the next generation they will be mixed, and the next generation they will be mixed, and so on and so on and so on. And all these things make that there are all the seeds, the sites here. There are some share the same history and are linked together. Okay, they are all the mutations are not independent at all. All these they depend on the rate that we call recombination rate. That is there are some part of the genomes that may break very often and then some that do not. And it's not homogeneous along the genome. And the thing is that it comes tricky. Is that if we consider recombination, there are, for example, here, the different part of the genomes they have different histories. Okay, some part of your genome, it comes from the father, and we come from the father or the mother, and so on. And so the part of the genome don't follow the same history. So before I talked about lineages, but actually in each of us, in each genome, there are many lineages. That is all the histories we share. Okay? And for example here, and this is linked with recombination. Okay? So for example here, we have this chromosome. If we in the population, there are different histories, 2 different histories, that in the extant chromosome, there is this part will have this history, this other part has this other history, because of the history of the evolution of the recombination. All these different parts have been recombined. So it makes it very tricky to look at the evolution of a genome as a whole in the population, because there are many histories and there are several, and they may on it there is all the selection and the drift of the alleles that make that some mutation will be kept or other will be lost, and so on. An important part about recombination is that when you have a mutation that is advantageous, it is good. I said before, this individual, that is, I have a good mutation, for example. I have a good mutation, then I will have many successors, and so on. But actually, it's not me, because the part of the genome, of my genome that has a good mutation will be split the generations because of the recombination, will be mixed with other parts of the genomes, and then this specific mutation will expand. But it's not this specific mutation, because it is not alone. It is in a part of, I said, a part of the chromosome that will be expanded. Okay? So for example, here, you have, for example, you have a good mutation here, and this good mutation will expand in all the population afterwards. But not by herself, but since it is linked to the rest of the segments around her, because of the recombination, because there are recombination tracks, there are parts. Actually, what we see is that this mutation here has expanded all over the population. Here we see the diversity, the level of polymorphism. So it means here that all the population has the same allele at this position, but also all around, because it is linked with the neighborhood. It means that when we look at the selection at a point here, if there is positive selection, there will be signs of what we could see as positive selection around, but there is not positive selection, only it is linked. That's the thing, and that's we call selective switch, is that there is some part of the chromosome that are carried by other mutations. Then be either that can be either what we could see for mutations that are positive things that we go to hitchhiking. I like the image of hitchhiking. Okay, so or either if the mutations are negatively selected, there will be some cleavage all around. There will be a positive selection, but just near a very negative one, so the positive will be destroyed, or the natural will be destroyed because they are linked to negative one. And that's what we call background selection. This means that the background is selection that is everywhere because even a natural one can be removed because there is a selectively mutation just near. So it means all this means that the sites we consider a genome are not independent at all. Okay, they are linked because of the lineage of their history, and they are linked because of the selection that may carry them. They can be the neighborhood. And finally, sorry, there is also, so I spoke about what happens in a genome, in the population. There are alleles, there are fixed, and so on. And then afterwards, I've shown you species tree. It means that there is a process of speciation. Okay, that we have, for example, we have one species, and then at a moment there will be two species. So, speciation, inside the species there will be sexual reproduction, okay? And we consider that when there are two species, each species evolves independently. Actually, that's what we consider usually, and usually we are very happy with this. It's not totally true. We know that there are some hybridization, some gene flow between populations, but there are always exceptions, there are always rules. There are many dynamics of speciation, but I won't go into it. There are many models, there are many studies. People are very interested into speciation. And there are the causes of speciation. You know, for example, the most intuitive one is you have one species, then there is a breakdown, there is a volcano, there is something between two species, and then each species goes its own way, and then there are two species. But there are many examples of one species that goes into two species in the same place, and people are very interested into what drives it. How does it work? Okay. How come that at first there were one species, animals, plants, and so on that could reproduce each other, and then slowly or not, poof, there will be two species. And these dynamics, they are very, very studied, and it's very important, and it can be studied at the level of population genomics, looking at the genomes, what is in the genomes, using, as I said before, the recombination tracks and so on, as the things I've talked to you before. So that's the main. I put some questions even before. So the main question in molecular evolution and in genomics is to understand evolution. So from the 19th century, from Darwin and some predecessors, mostly Darwin, many questions about evolution. How have the species evolved? What is the dynamics of evolution? What is the importance of selection, as I mentioned before? How does speciation occur, as I just said? Is evolution gradual or punctuated? It means that are species evolving slowly like this, or there are sometimes some bursts of species, of evolution, because of many, and which features, for example, what ecological features may have an influence on the evolution of species. And what is the importance of population structures, the breeding strategy, the reproduction strategy of animals, plants, and so on, the sexual system, or is it linked with evolution? There are many, many, many questions about this. And from the from the 70s, there are more and more genomic sequences. There is an exponential increase. So, for example, more and more this from the 2000 because of what we call the new generation sequencing. And so there is an explosion of research on genomic evolution because, as you may know, the people haven't waited for genomes to study evolution. First, it was more on the field, and there was Mendel with his peas and so on, and now we have genomes, and so there is an an explosion of modeling, of methodology in evolution, because there are many data. Okay. And so there are mostly two approaches. That is, there are population genetics and phylogeny, as I will explain. So what does it look? Broadly, it is that population genetics, so you have 1-2 species, in which we consider the whole population and all the polymorphisms you see here, is polymorphisms or not, that could be involved in these populations. And phylogeny, totally different, is that you have a tree, we have one individual, we can say per species, one genome, but we consider many species. Okay? The scale is totally different. The thing is that in both approaches, the analysis use many sites, because they will bring much information. So first in population genetics, so from the 20s, the founders are Fisher, are done right in the 20s. They made a lot of the very impressive work on theory. Theoretical works on population genetics are still very relevant. So most of their work is a top-down modeling. They say we have given an ancestor, or does it evolve, and then we can predict how the alleles with the frequencies of allele will evolve, or all this stuff. And so from this, there are huge number of statistics that are used by people looking at population genetics. They are either intraspecific, that they will look, for example, at genetic diversity, looking, for example, position along the genome, if there are alleles that are very rare or common, what is the frequency, what is the distribution of the alleles. So the difference between the rare and the common variants, and from inside species, the tracks or recombination tracks, as I mentioned to you, the places where they have been broken, the length, the repetition a lot along the genome, and so on. As well as what we call the linkage disequilibrium, it means that looking for traces of selective sweep, the places where it could be displaced, there may be some positive selection and so on. And they will also look at interspecific analysis between populations. So, for example, to look at polymorphism that would be shared between species, that is, there are change that is the same in two species. So why is it old and so on, or distinct, and also the different fixations. In one species there is one allele that is fixed, and the other one is another allele. Why, or is it, or have been up to this and so on. So the methods in population genetics, they're mostly based on polymorphism. They look at sites that are variable inside the species, or inside two species, but rarely more than that. So I will stay very quickly on this one because I won't have time. So first a very simple, actually very simple, at base very intuitive and test of selection. It's what's called the Mcdonald-Cadman test. It is only, I won't come into the formula. It's only to say if a part of a genome is under positive or negative selection, and we just look at the number of different patterns, either if the proportion of divergent sites, the sites that have different alleles, and polymorphic sites that are shared, okay? And we can have the ratio between the sites that are polymorphic and over the sites that are divergent. And we can look at the ratio between different categories of sites, and their index is only we make the ratio between the sites that are non-synonymous over the sites that are synonymous. We could expect that if there is no selection, this ratio equals 1, because there is no difference between the dynamics of non-synonymous sites and the dynamics of synonymous sites. But if this ratio is away from 1, there is different behavior between non-synonymous and synonymous sites, and that will be interpreted as at first it would be interpreted as selection, either negative or positive. But afterwards, people do, it doesn't behave like this, and go further into the model. But that's another question. Another, as I said, another use of in population genetics as a motion is looking at what we call the haplotypes, the name that would be the part of the genome that were mostly on recombination tracks, and look at the history. Okay, for example, here there are we can from here different populations. We can have some. We can look at different parts of the genomes and say there are some parts that have a different history. For example, here you can see there are the history in blue, the one in gray, the one in green, I think. And so, looking at the different parts of the genomes, we can see different genealogies and then understand how these populations have evolved, the dynamics between the different populations. Okay. And so, from along this, there are also many models, and there are many statistics using these different genealogies, these tracks, to infer the recombination rates, the incomplete lineage sorting, so at which point there can be different kind of topologies, different kind of histories, and we also use tracks of link sites to infer selection, as I mentioned before. That's very used in population genetics. And finally, there is another approach. So the approaches I mentioned before, they were mostly based on statistics, started from top-down model, and there is another approach that is more bottom-up, that's called coalescent approach, that is started mostly in the beginning of the 80s, that is given several individuals now to infer where they come from and what is their ancestral. Okay, that's the coalescent, is to infer from the extent, so it's a different approach, given that's what we use, for example, for different women, given different genomes, different persons, we want to infer their genealogies, their family relationships, and to come up to their ancestral, to their most recent ancestral. Okay. So this is there are many works in using coalescent approach in population genetics that are usually here. We have 3 species, but usually they are inside the species. But as I will mention later, there are more works on what we call multi-species coalescent. So at first the models were but neutral, so and no recombination. So there was only the evolution is neutral, and there is no split. We only consider blocks, and then more and more models using selection and recombination, but they are very studied as well, and sometimes very theoretical too. And on the other part, there is phylogeny. So phylogeny, as I said before, it's another point of view of evolution. In this point of view, we forget everything about populations. So we don't consider polymorphism. The idea, the hypothesis is that polymorphism doesn't remain long enough. There is only, as I said, there's only bubbles, small bubbles, that are removed, so we forget. Okay? Recombination happens often enough. That means the chromosomes are all broken, very often. So all sites are independent. Okay? And we consider only 1 sequence per species. It's with the link of not only polymorphism, but the fact that we don't consider, for example, the different lineages, and so incomplete lineage sorting, we forget there is one evolution along a tree that is a species tree, and that is totally coherent with the evolution of the genes that we use for the inference. Okay? So here the idea is that we forget everything about population, we have this. So at first, the first model of phylogeny is used. On the contrary, we forget everything about population, but we will infer, we will look at the evolution of species. Phylogeny has been used for until the last unique common ancestor, very, very deep phylogeny to understand. It's very, very efficient works. It's very many powerful tools on this. And on the modeling point of view, the first models of on phylogeny were made by Jacques Cantor. It was in '69, and so they were only looking at substitutions. Okay, so there is no mutation anymore. There is no polymorphism. Only substitution. Everything is fixed. Very simple from this point of view. At first, there was no characterization of substitutions. For example, the first model, we are here the 4 letters A, C, G, T, and the arrows represent the rate at what is it fast or not to change from A to a C to a C to a G, a E, a T, and so on. And at first there was no difference. Actually, it's more or less like the in population genetics. I said before, we don't care at all about the pattern of substitution. Okay, perhaps because we don't have the data. But the thing is that it was not the point. Here we consider rates between letters. That's a computer scientist point of view. And, but actually it didn't fit at all, because we noticed, for example, here, considering this model, we should have, for example, in all genomes, the same number, the same proportion as A, C, G, T. Looking at the data, the genome, no, it's not good. It doesn't fit at all. So afterwards, people made more and more sophisticated model. So and it was the same, for example, for models looking at the evolution of proteins. The first model of Jacques Cantor was the same, equal rate between all the amino acids, so we should have the same frequency of all amino acids. That's totally false. So now the approach is not mechanistic as it is in population genomics, because it's based on models, very elaborate models of evolution. Here, the approach is more phenomenological. Okay, there are substitutions, the models don't fit. Okay, then we fit the model to the data. We don't care about the mechanisms. We don't, perhaps later. No, we have to fit. Okay, the point was there were many goals from this. For example, to build up phylogenies. So it is here that the approach is mostly phenomenological. Okay. We have a model, we have the data. We have to change the model to fit the data, but with parameters or not. For example, here on proteins, there are many the models that are used. That was in the '72. It was very, very short after the first models. They say, okay, they fit the model given what is observed as protein frequencies. Okay, the model, we use this one. That's it. On nucleotides, since there are less parameters, can perform data-specific optimization, that we can have a model that will be fit for this gene, or this gene, or this gene. And so the first one was for Kimura in 80, which is only the rate of transition, but after there are many more models. There's been a modeling more and more elaborate. That is mostly data-driven. That is, the model were not, it was not top-bottom approach, but the inverse. We have the model, we have to feed the data, oh, it's fine, and then we discover a new feature. Actually, on the phylogeny, in phylogeny, people in phylogeny discover very interesting features in the dynamics of substitutions. Okay, but very totally apart from the question of population genetics. It's not the point, not at all. And for example, for substitution, for selection, if I come back from selection, the first model looking for selection was made by Young and Nielsen in 1908. So there, as I mentioned before, the one main part of the genome where there is a selection is inside genes, and by looking the difference between the dynamics of synonymous and non-synonymous sites. And for this, it was very convenient. For this, the Guéguen himself proposed a model that is made on substitution of codons. So here we don't have 4 states. It's not a long ACGT. It's all the 64 states. Actually, we refer 3 states because they are the stop codons. And so given this model, there is one parameter. For example, there are many other parameters that are called usually omega. That is the rate of non-synonymous substitutions over synonymous substitutions. Okay. So there is, we put this, the idea is that when there is positive selection, the non-synonymous substitutions are more frequent than the synonymous one, that will be other 1, and the inverse it will be below 1. And so it's quite efficient. Okay, it's a model that works well, and from this, this model has been expanded to allows now site-specific estimates, branch-specific estimates, and so on and so on. There are many, many modeling. But the thing is that this model, so there is a, okay, it fits on the idea of substitution, but it doesn't fit at all with, it doesn't fit. We can have questions because, as I mentioned, selection is defined in the matter of mutation and then fixation. It's a definition. Selection is defined in population genetics, not in phylogeny. It was only okay. We want to find it. We set up a parameter, and so now there are been new models that have been proposed. Actually, it's the same date. That was called mutation selection models that want to model evolution of genomes given mutation, selection, fixation. All the dynamics I described to you before, but in the context of phylogeny. So this model want to be more mechanistic, and the practical problem is that there are usually site-specific fitness. They want to describe this fitness that is on each site. Okay, as I mentioned to you, the fitness is the ability of an individual to reproduce and so on. Here we define the fitness on sites. I don't know. Actually, I don't know what it means for me but on a biological point of view. But in the modeling, it's fine. But at the same, there are site-specific fitness, and there are many parameters. So these models are very complicated to estimate. But they do many. They are very interesting also, and they are very interesting in the fact that now they tend to break. I should say the omega parameter, the selection parameter, is something that will be where it is the mutation part, what is the selection, what is the fixation part, and so on. Okay, and to involve, for example, in this modeling, the idea of population size. As I mentioned to you, the size of the population is very important to assess the drift, what is the strength of the drift, and so on, that it will be included in this modeling. Okay? So the idea is now, okay, we have very efficient models, given certain goals. Okay? Now, if we want to come back to understand really what happens, we have to not break, but we have to develop this model to have something more mechanistic. But even with those models, we have still the limits of phylogeny, as I presented before, because of the hypothesis we set. First, there is no polymorphism. Okay, we forget anything about polymorphism. So, since all the mutations are seen as substitution, there will be, even if we see a mutation, we see it is a substitution, so there is less. Each mutation it is as if each mutation is fixed, so there is less selection because they are more tolerant. And as well, the speciation are seen as instantaneous. Okay, there is on the tree there is poof like this, one species, two species. So just a little spot here, and there is no all the dynamics linked to speciation, and so there will be no, for example, no incomplete lineage sorting. The fact that some lineages don't follow the species tree, and so we don't model any dependency between the species that are very near, like, for example, gene flow and so on. So the sites are considered as independent, as I mentioned before, which is usually false. So we forgot anything about the link selection. That we would say, this gene, this site is under positive selection, but the other one not. It's very strange. Okay, so and there are many modelings there are many questions about what means to be, in that case, in a gene to be linked, to be on a positive selection or not. And we don't exploit, in phylogeny, I say we, but in phylogeny, people lose information about recombination tracks. We don't integrate any, that's the thing because sites are considered as independent, so all the region of the neighbor information is lost. Okay, whereas in population genomics, this information is very important. Okay, the recombination tracks are very important. So the challenge here for phylogeny is to import information from populations. Okay, to make the link to say, okay, there are methods in phylogeny that are very efficient, but especially if we consider phylogeny for recent phylogeny, won't be very good because we have made a lot of hypotheses that are wrong, and we have erased a lot of information. Now we have to bring back this information into phylogeny. So for this, there are mostly, perhaps, I discern two approaches. I'm sure I forgot some of those, but another first approach is to First is to model to manage polymorphism. Okay, we have polymorphism. We want to integrate it into phylogenies. A first approach is to, like the model in Wilson, to say, okay, we know we have models, and we know if here we had one single allele in the position, what we expect as distribution of alleles at the end of the leaf, at the end of the branch. So we will use population genetic models only on terminal branches. What we call terminal branches are the branches that are when there is only the species, there is no shared polymorphism, there is no ancestrality. It's only at the tip of the branches. We will set a specific modeling. Okay, that's not very tricky. We can make it quite easily because there are good models of evolution here. But in that case, we consider there is no ancestral polymorphism. Okay, there was one, for example, on one point there was one nucleotide at the top of the branch, of the terminal branch. Now we have polymorphism. Okay, we integrate polymorphism to have better estimate of what was before, but above, it was as before, one lineage and only one state. So we don't consider any ancestral polymorphism, and we don't consider any problem with the phylogeny. Okay, there is no incomplete lineage sorting because we consider one topology. That's it. Another approach is like POMO. It's a phylogenetic aware MO. I can't remember. So here the difference is that we model the dynamics of the allele frequencies. For example, here we consider that the population, we split the population in beans. They can't model population with many individuals. It's much too complicated. So for example, they will consider that here there will 10 beans in the population, and for example here there is a population that 10A, and then perhaps before there was, if it was 10A here, just before there was 9A, 1T, and before 8N, 2T, and so on. So they model the spread of alleles in the population. Okay? In that case, they will model the evolution of distribution of alleles all along the tree. It will be fine. With no, in that case, they will model incomplete linear sorting, because they will manage all the polymorphisms. But the thing is that here, as you can say, for example, in this model, they have, I should say, they have more than there are 60, something like 60 states. 60 states. On the computational point of view, it's fine. Not much more. Okay, it becomes a bit tricky. So now, so they are limited to nucleotides. If they want to model codons, they would have hundreds and hundreds of states, and computationally, it's not feasible. It's more too complicated. Okay, so the problem is there. The idea is very good, and but up to now they don't have, but it's still limited. Because of the computation, it's limited to nucleotides. Okay. So now they use the model selection in this model, but it would be like the average selection of all nucleotides and not specifically on synonymous and not non-synonymous nucleotides sites, because they would have to handle codons, and this it's too tricky. Computationally, it's too difficult. Another approach is no. Okay, another approach is called the multi-species coalescent. As I said to you before, the coalescent is the bottom-up approach that, given extant genomes, will try to infer the ancestral, their order genealogy, where they come from. And the multi-species coalescent is the same as before. It's the same, but this time we consider different tubes. Okay, different populations that are mixed at a given point with several parameters that would be here the age, the date. Here there are many parameters, and they will look for all the scenarios that can explain the genomes, the extant genomes. And with the concept, there is this time of tree of populations. It's not species tree; it's tree of populations. Okay. And to have inference of the past, the evolution dynamics. There are many formulae in this kind of approach, which are quite interesting. The thing is that since they want to model the limit, I see, my point of view is that since they want to model the past of all possible scenarios in the past, even if they have combinatorics approaches, they are limited with the fact that of the number of lineages they have to consider. But nowadays, when there are sequencing projects, there are hundreds and hundreds of genomes that are sequenced. So, and they can't, in their approach, it's not possible to handle with this specific, when they look specifically at the lineages, to handle hundreds of genomes of lineages. Okay. So in that case, the approach that would be more like the previous one, when we consider allelic frequencies, is more will be more helpful in that case. More computationally, we can do more stuff with this. So here is a nice example. I just put some example of applications that was bought in Nature 3 years ago. That was, for example, to human history. You're using this approach of multi-species coalescent using several genome of extant human here and archaic humans, like Denisovian, Altai, Alcideron, and some Neanderthals. They have estimated the gene flow, how much of the genes that are, for example, in the European, Chinese, and Papuan here, how much part of the genes have been come from the Neanderthal genomes to our ancestors. Okay, and for example here, so here between the dynamics of the different archaic genomes, here it was the population size. And so using this approach, multiplaces coalescence, they have managed to reconstruct. It's a very, very impressive work. It's very, very nice work. They've made it, they've made it in, for example, this example is with human history, but there are many works as well in mostly human, we should say, because there are many, many studies about humans. But in every part of the world, we're looking to understand how the human populations have migrated, have linked, and so on. That's very, very nice works that are, and many are based on this kind of approach. So, as I said before, it is not possible to look at all the histories, and so we can take advantage of the fact that there is track, as I mentioned before. Okay, there are genomes are not, the sites are not independent. There are tracks, so we can use the tracks to infer the histories. So this is another approach that uses coalescent approach. But here the models are much, I would say, they use less data. Here, for example, we use the point was to infer completely in a shortening between human, chimp, and gorilla. So as you know, the species tree says that human and chimp are closer than to gorilla. And so what the people have done is to consider 4 different scenarios here. So there is the usual one. Here the scenario, we're looking at part of the genomes where the human and the chimp and the gorilla are linked, but very, very long ago, more long ago than here. Here, for example, the chimp and the gorilla are closer than to the human, and here the chimp and the gorilla, no, here the here the human and the gorilla are closer than the chimp, and here the chimp and the gorilla are closer to the human. Okay, so there's part of the genomes where the evolution is not specific, it's not like we expect in the species tree. Okay, and they have so they have used what is called the hidden Markov models that are they looked at segments, part of the genomes where there is this kind of genealogy, this kind of the genealogy, and this kind of the genealogy, okay? And not each site independently. So they used, and it is a probabilistic approach, and so from this they estimated the different kind of scenarios is the same color code here as before, okay? Usually it is the usual, the expected genealogy here in this color. I can't say which the color is it, but it is the human chimp scenario that is more frequent. But there are some places, for example, here there is a place, there is on this chromosome there is a place where the genealogy is human gorilla are nearer than chimp. Okay. But here we can, so we don't have, didn't use any recombination track of population genetics, they only use one sequence, okay? One genome of human, one genome of chimp, one genome of gorilla, but with this model, they say, okay, on one side you couldn't have said that this coalescent is, that scenario is better than this one. It was, there is not enough information. But there are several sites that all go towards this scenario on this track. Okay, so the very nice, nice work, because the idea is not, okay, we don't have the information, it's very too complicated to look at all these tracks and to do like we could do in population genetics. We just consider one, but we use the fact that there are tracks. And so we will agglomerate, I should say, the relevance of this scenario and say, okay, here we have this scenario, here this one, this one, this one. Okay. Well, so it's an approach totally different, that is more, that is probabilistic in the sense that we don't look at all these, the patches we have, but we don't look at all the patches to reconstruct the history. But in the model, we consider that there are patches, and then we just use one sequence. but we recover patches. Okay, that's very nice. Sorry. Sorry. Okay, with time. Okay, sorry. I will just. Okay. Yeah, it's a little bit long. So it will be other approaches using graphs. Okay, we go faster than this. That will be new approaches when we consider, instead, it's the same idea, instead of looking at all the scenario, we mix the scenarios inside graphs. And, okay, so it is a conclusion, I was not here. So the point was how to use population evolution in interspecific framework. So there is the coalescent framework approach that focuses on lineages. That's very accurate modeling, but can be quite heavy to handle. With no, and with removing lineage consideration, we use allele frequencies. So on bottom-up modeling. What is still to be done is my point of view is to, well, there are a lot of to be done, but there are many interesting things, is to compute the evolution around evolution graphs that will infer hybridization of different less distant than not only trees, to model, to include background selection in hiking, and in all I presented to you, the idea nowadays was to include population genetics into phylogeny. But I said to you before, phylogeny, as we have, there are many interesting stuff in that describe quite well long-time evolution in terms of substitution patterns, direction of substitutions, and this information is not, as far as I know, that I'm mistaken, is not included into population genetics nowadays, you know. So there is still, the thing is that we want to include population genomics information into phylogeny, but the other way is not, as far as I know, is not very looked for. Okay, thanks. Sorry for being too long.